Thank you very much. Really appreciate that introduction. And, um, and thank you all. Happy Thanksgiving back in the United States. It is a day of gratitude. That's my personal favorite holiday. And I'm very grateful and honored to have been invited to speak to you all. And I'm very grateful to our hosts who've been so kind and generous and welcoming to us all. And I'm very grateful to all of you who have come all this way to share all of your knowledge on jellies. I am a curator of what I think is the largest collection of jellies in the world, and I'm often introduced as a jellyfish expert or referred to as a jellyfish expert. And these last 2 days is very humbling because it's a nice reminder of how much I don't know. And so thank you for all sharing. So I am going to talk today about the pace and scope of characterizing jelly diversity. And the first little part is a bit of a point of view, and then the end is some more current research. So by jellies, I'm talking not just about scyphozoan jellies, but the cubozoans, the hydrozoans, and in addition, the staurozoans, as well as the comb jellies. And I should have added salps and pteropods and other gelatinous organisms to this as well, had I been thinking, but I've been convinced for sure by some of the comments that have been made during the conference that we should not neglect the other jellies. So by characterizing, what am I talking about? So documenting basically all the information that could be useful about a particular species. Of course, there's morphology, macromorphology, micromorphology. Ideally, we know something about the variations of these morphologies, and we want to know the morphologies for all the different life stages. And then linking that together, information about how they develop, how the morphology changes during ontogeny. And underlying all that, of course, is information that's encoded in the genomes, the nuclear genome, the mitochondrial genomes, the genomes of symbionts, so associates are incredibly important. And then likewise, we want to understand how those genes are used by the organism dealing with stress or any sort of the functions. So we have all of these things have been illustrated by the talks that have come before this. Of course, ecology, habitat to prey, predator, parasite, and other associate relationships are incredibly important. The behaviors matter to us. We're studying jellyfish blooms in this, or focusing on jellyfish blooms in this conference, and so behaviors are incredibly important for that. You must be interested in phylogenetic position, so you would want to know this about every species, is how are they related to other species. That way, in the absence of information from that particular species, you can make predictions about characteristics. Geographic ranges, of course, are incredibly important to know, and then population structures. And we've seen some nice examples. I'm thinking of Connie's genomic-based work with Nemiopsis, which not only shows us population structures as they currently exist, but gives us information about recent time and recent events, even invasion events. And of course, we can all think of a whole lot more. I think our grand goal is to know pretty much everything about all jelly species, everything that could possibly of interest, of impact. And the key step to a key step in the process of understanding this for all jellies is being able to link the information together and the knowledge tied to something. So one way that we know species is by describing them and naming them. This is a species accumulation curve for the comb jellies, and at present we have about just under 200 currently accepted species, and 19 of those have been described since 2000. So in the last 23 years, about 10% of those. I just quickly looked up the number for salps and for the pteropods, and those numbers are 6% and 5% respectively. So in the last 23 years, we've added that. So there are hindrances to our progress in knowing diversity in this way, naming and describing things. For comb jellies are notoriously bad preservers, so they don't wind up in museums very much. They can break down very quickly after you collect them, so you might recognize something as new, but then not be able to document it fast enough because many of the diversity is in the deep sea. It's relatively inaccessible. And of course, for all of these groups, we have a relative lack of taxonomic effort. We don't have enough taxonomists. We know there's a lot of diversity out there to still describe. But is progress in characterizing ctenophores really that bad, that we've only make incremental progress? I'll argue that we're getting there faster. And the numbers are pretty similar for the medusozoan groups. Also, 10% of storozoans have been described in the last 10 years. The numbers for cubozoa are the exception, where fully half the species have been described in the last 23 years. And I talked about this way back in Barcelona, that there was this big uptick in cubozoan systematic work, and it has continued. So it's really nice to see that that pace is continuing. There's a lot of focus on this group because of obvious reasons of their biology. And then there are the numbers for scyphozoans and hydrozoans, 14% and 21% of species in hydrozoa have been described in the last 23 years. So that's a relative anomaly. But then what about this problem? Sometimes we get described this problem of known unknowns. So we tend to accumulate in the molecular genetic literature a lot of species that have informal names. So you think of all the Aurelia species or genus species X. So those are accumulating in the literature. So we are accumulating knowledge about species, but we don't have a formal species name for them. And then there are a bunch of things that we know by what they look like. So these examples from ctenophores, and these get sort of pet names. Tenoceros, this is the sidipid comb jelly that has these horns. Benthotina is a name you sometimes see. Liver lips is another informal name. And these names can also be used to link up information. So we know that the first Aurelia genome was published as Aurelia species 1, Cerulia. And, but still, those, if we're careful, those names can be used to link up diverse information. And there we've even seen in the literature Aurelia species X described from some bay or whatnot, and its behavior. So that can be used, but they're because they're informal, they're a little more vague, and they tend to be a little less certain. But they're still an important thing. So in the context of this whole question about how are we doing, I have been thinking about what is the ideal species description, and it's just a list of actually knowing all those things that I've already ticked off. So I have a jellyfish, Scolianema. I discovered it in my lab, or actually Rebecca, my friend Rebecca, discovered the polyp. I want to learn the life cycle, so we've been able to raise it in the lab. I've been working on the genome and sequencing it, and of course we would want to know, send it out for transcriptome studies, so what genes are doing, and there's gene cascades are being worked on in this group. I know that I can raise it all the way through its life cycle on Artemia alone, unenriched Artemia. So that's important information, but I don't have information on its natural diet. But this is something that would be that we would want to know for this organism, its natural diet and its natural predators. We have information on its phylogenetic position based on the genetic data, and I know that it comes from my little 55-gallon aquarium that I keep in my lab, but I want to know where it comes from in the world. And so I'd like to include that information if I were to ever describe the species. And then ultimately, it would be nice to know its population structure across this whole thing. So every species is this multitude of stories that we've been telling through this whole conference. And of course, they don't just happen when we collect them. They have these histories to them, near histories. A species might arise in one part of the world, but spread to others, and also deep in time. So I wanted to share a couple of examples of less than ideal species descriptions that I've been a part of. This is the story of the two-handed ctenophore from Puerto Rico. And back in 2015, this was observed. There were 3 separate observations. The lead scientist on this trip was my colleague, Andrea Quattrini, and she gave this the very first descriptor. She called it beautiful. And because this was seen with NOAA Okeanos, they have a very rich telepresence process where there's lots of scientists back at home that can watch live and comment. It was also seen by my colleague Mike Ford, who called in and then was on the live broadcast. And he also said, wow, that's beautiful. But then he went on to describe its morphology, noting the general sort of body shape, the two tentacles, and referred to it as a sidipid ctenophore, very general. But we decided we wanted to write a paper about this observation, even though it was only recorded on video. But at this time, I had a very talented scientific illustrator working with me in the lab, Nick Bizzaio, and he started sketching this out, and I realized we were getting way more information out of the videos than I expected. And I thought, well, maybe we can actually name this species. And so we did. We got the name Duobrachium sparksei. And instead of a physical voucher, we have high-definition videos that are also, they serve as the holotype. So these videos and our description are what equals this species. Less than ideal species descriptions, but I think which move the field forward. My good friend Jonathan led this study on Aurelia, and we all know that Aurelia, some of the species have definitive morphology that's easy to tell apart, but many of them have accumulated in the literature as species X because they are difficult to tell apart. And we can do morphometric work, and indeed, Jonathan was really interested in trying to connect morphology to these species descriptions, but in the end, it was more relatively easy to differentiate these lineages based on the genetic data than it was on morphological data. And so hatched the idea that maybe we could describe them using these characters as the diagnostic characters. And so many of these species that we created in this paper are based just on genetic data. And that's a less than ideal solution, but it, I think, moved the field forward, solidifying names that can then be used to join up information that are gathered by others. Now I want to just turn to some work that's going on in my lab with Nick Bizzaio. I'm going to focus on ctenophores and ctenophore biodiversity characterization. So we all know about the phylum. There's about 200 species. Comes in a lot of different body forms, the lobate forms, the burrowid forms, the tentaculate forms, and the benthic ones. So one of the groups that Nick is focusing on are these burrowies, and they're basically living stomachs. The majority of the body has a vast gastric chamber. It's pictured in gray on the right. And the system that converges at the apical end of the animal branches into 8 canals. 8 canals that go under the team rows, and from those canals we have these diverticulae that form. So burrowy literature, Nick's been going into the literature and looking at all the different names that have been suggested for different burrowy species, and he's found accumulated about 300 different species descriptions. And then his assessment, after looking at sort of obvious synonyms or taxa for which the description is too low quality to make anything with, so species incorrenda, leaves about 16 different burrowy names. But people are collecting and sequencing burrowy around the world, lots of different groups, and they tend to always get identified by these 6 names. Burrowy abyssocola, for instance, most burrowies that have deep red pigmentation will be called Burrowy abyssocola, Burrowy cucumis, gracilis, etc. You know these things. But what are the genetics telling us? So basically, they are far more diverse than 6 and more diverse than 16. At present, we're doing species delineation methods that indicate that there's about 20 different of the burrowy lineages, distinct lineages. Now, because Nick is has a great eye and is a keen morphologist and is able to transfer that morphology to images that people can understand, he's been drawing the specimens that we've been using to collect data, or that other people have collected data. So they come in a variety of sort of shapes, right? But these animals are very squishy, and they can change these shapes in very short periods of time. And so He's looking at other characters, and in particular, he's been focusing on the canals and the diverticulae. And so you can see these are cross sections of the canals with how the diverticulae come out from the canals. And he's able to find unique patterns that associate with some of the genetic lineages. And so this is a possible character tool. He's looking at other characters, of course, like the macrocilial teeth at the front ends of the burrowies. And this is allowing us to make first inferences of how these characters have evolved within the group of burrowii. So you can see with a simple tree like this, we might infer that the type 1 teeth are the ancestral condition within the group, and that these type 2, 3, and 4 have evolved independently. So the other group that Nick is spending a lot of time focusing on are the platytenids, the benthic ctenophores. So these animals, they do have a pelagic part of their life cycle. They do have the combs and when they're juveniles, but they settle down onto the bottom. All but one species lose those combs, and they settle down on their mouth, and then they have the 2 tentacles that they deploy up into the water column in order to catch organisms. So he's roughly dividing these into sort of shallow water groups and focusing on them, and as well as the deep water groups. And this is an example of several pictures of Coeloplana, which is a quite diverse genus found in many shallow water environments. And these species have been differentiated in the literature and described based on basically 3 different characters: the arrangement of some papillae that are on their aboral surface, the host or substrate that they're found on, and then also the coloration and the patterns of the pigmentation across the body. So, but how do these characters relate to the genetic data that's coming out on them? Well, it turns out that these 3 features do a good job of sorting out the different species. So unlike the burrowies, there's quite a good correspondence between known and described species, not perfect, and these characters. So these genetic data reinforce the morphological delineations. He's also working a lot on deep water platytenids. So at present, there are only 2 species of platytenes that are described from greater than 200 meters, and that's Lyracteus imperatoris and Gelfiella tristoma, described from very different parts of the world. But we know from lots of ocean exploration that is associated with high-definition video that there are quite a number of species that are down there living on glass sponges and octocorals typically. So we know that there's a lot of known unknowns. There are species there to be described. And as we've been accumulating the genetic data, in fact, it's true that almost every different one that we've been sequencing is a distinct lineage, one from the next. Now for this work, Nick has been doing computed microtomography, so micro-CT, and unlike most comb jellies, the platytenes are quite good at preservation. So these are preserved, and then they're amenable to being put in the micro-CT and studied. And then Nick, because he's such a good illustrator, is able to color code the different parts of the ctenophores, these specimens here. And then he's making drawings of these new species based on the micro-CT data. So we expect to describe a lot of these species in the near future, and they will come with sort of morphological descriptions coupled with the genetic data that differentiates them. Now I want to turn a little bit to the method that we've been using to generate genetic data in our lab lately. It's not a particularly novel technique, but it's called genome skimming, and we've been doing it at a large scale. We've been doing this for thousands of fish species. The U.S. wants to have voucher-based genetic data for every species that lives in U.S. waters. We've been doing it for sea stars, and I've been doing it for hundreds of sponges. So it's really simple. You just start with a DNA extract, and this does not have to be a really high quality DNA extract, because one of the very first steps you do is you shear this DNA down to about 400 base pairs, so an average of 400 base pairs. Then we pool those, we tag them, pool them, send them out to sequencing on very new technology sequencers, NovaSeq sequencers, Illumina data, paired 150s, and what we wind up is, then we get 10 to 20 million reads per sample out of these. And then what we do is we just basically assemble them. We create kind of a poor quality whole genome assembly. And you see that in the bottom right there. That's basically just seeing where do these sequences line up, right? Where do they go together? And that gives you a list of contigs of various lengths and various coverage amounts. So that's the amount of data that's sort of behind them that lines up underneath them. And then with that list of the contigs, we're able to search through that bioinformatically to look for, so where is the cytochrome oxidase 1 gene? And that almost always comes out on a long contig that corresponds to the mitochondrial genome. And we can look for other data in there. So we've been mining it for the ribosomal repeat units, which is 18S, ITS1, 5.8S, ITS2, and 28S. And then these data contain a lot of other information. There's microbial data in there, so we've been mining the sponge ones for microbial data. The ctenophores are full of photoproteins. So evidently, photoproteins have a lot of tandem repeats, and the types of data that are easy to get out of this are data that are abundant in the genome. So the mitochondria, we have lots of mitochondria, the ribosomal repeats. Oh, advance. So here's what we've been getting we've tried about 107 ctenophore samples, and a couple of those were non-ctenophores. 5 of those failed, but for 90 of the 107, 84%, we're able to retrieve the complete or the very near complete mitochondrial genome. And we have 10 partial mitochondrial genomes, and they all include the commonly used COX-1 barcode. And of those 107, more than 90%, we can get the complete ribosomal repeat unit. These numbers are very similar for what's our success rate in sponges, sea stars, and we're doing Medusae as well. So this technique is relatively inexpensive too. So we can do it for about $40 per sample, which is not that much more than trying to Sanger sequence 2 or 3 markers. So of course, there's lots of other information that we can get out of that. This is a backbone phylogeny from these new data across the ctenophores. And in addition to sort of supporting the taxonomic work that Nick's doing in the baroids and platytenes, we know that there are people who are working on some of these taxa that have these known unknowns like Tenoceros or liver lips, and we're able to place them in a phylogenetic context and provide this data to the people who are describing these taxa, which we will do. Also, I want to mention that through this genetic characterization of species, these make them accessible to being found in metabarcode libraries. So these are cytochrome oxidase 1 metabarcodes that you can see directly link a sequence to this in this case, a ctenophore Euramphia vexilligera, I think. And this is the ctenophore that inks, and we know more about its biology, and so now we know the places that this can occur. These metabarcode data and metabarcode occurrences are going to be put into GBIF, and so GBIF is working in a way, the Global Biodiversity Information, and this will rapidly expand our knowledge of what organisms are found in what parts of the world. There are other one of the reasons we did the genome skimming is that many different groups are targeting metabarcodes of the fish people like 12S or some part of 12S, cytochrome oxidase 1. This is a case where another guy, Luke McCartan, who's a PhD student at Lehigh University, he's working on corals, and he designed some coral-specific primers for 28S that he thought were coral-specific. And he told me they had a bunch of non-specific amplification. And so I asked him if I could see those primers, and they're perfect matches for the ctenophores. And so he has been generating lots of ctenophore data that he didn't know about. And then on the left here, you can see some of the ctenophore barcodes that are aligned against our voucher-based 28S sequences. And so this will tell us what diversity we're missing, where we need to collect more stuff, and pretty excited about where this is going to take us. So we've heard a little bit about eDNA, and I'm in the museum, and the museum may become a place to voucher eDNA samples, and I would suggest that you consider vouchering at least some of your samples, putting them in a museum so that they can be used by later researchers for other technologies. And in the presence of other knowledge, you may be interested in species X to do sort of like quantitative PCRs out of that, but those samples are a snapshot in time and space, and you're never going to collect that sample again, right? You're never going to go to that place again at that time. So they have lots of stories in them because they're holding lots of species occurrence status. But they can only be identified if we have reliable voucher-based barcodes that you can go back to and look at the original specimen and say, no, that was wrong, you know? So we need to have a system in here where we can fix these things. And of course, eDNA is being deployed by lots of people. I work for NOAA Fisheries, and in NOAA Fisheries is planning on collecting tens of thousands of these samples for their work just in US waters. So I know that this is going on in lots of different places in the world. And so the genetic characterization combined with the key is the museum specimen that someone can go back to, even if it's high definition video or good photos in the lab before the animal fell apart, because eDNA is being used to address all sorts of fishery management questions, and to influence decision making. And with that, I would like to thank you very much for your time and attention, and